Okay, I think it would be fair to presume we're moving on to, per se, a different type, or not a different type, but a different unit, okay? One of the things, probably one of the bigger take-home concepts from last chapter, I just happened to see, it was maybe a strange post on social media, but it was talking about two different types of soldiers from Australia and from the Marines as well, and they're doing a workout that would pertain to what we had discussed last chapter, okay? And that is of the following. You had two types of soldiers, the Marine and the one from Australia, and they're both holding a 35 maybe pound weight like this, okay? So when you do this, it's pretty easy to start out. This is pretty simple. Okay? But eventually, what's going to happen? They're going to get tired because you're not producing enough what, do you think? It has something to do with oxygen and the mitochondria, but it's a different type of, what do I want to say, process. Because how does the body produce energy the most efficiently? Through what process? It's a cycle. The Krebs cycle. And the only way that Krebs cycle can continue is if it has enough what? Now you can say that. And then where's that oxygen taken in and ATP broken down is where? See, those two are related, okay? The mitochondria and the ATP. Eventually, if you see someone like this, they're probably going to start doing this, and that is why. You don't have enough oxygen to go around, but you still can produce energy to flex these muscles. It's just not very efficient. That process starts with a G. Glucose is broken down in this process. Glycolysis, okay? And if you remember the numbers, I could hold it up there a while, but yeah, I don't want to harm this noggin. So, all right. So with that, the body still can produce energy, just not aerobically. It does it anaerobically in glycolysis. And if you remember the number in that diagram, we said in the Krebs cycle, it spits out how many ATP molecules? Right? Not 7, 34. Pretty efficient when you compare to glycolysis, which spits out how many? 4, yes, but it takes 2 to run the process, so you only get 2. So you're half right. Okay, so moving forward then, we begin talking about types of, I think it says molecular genetics, I believe. So let me go ahead and bring this up. So just like anytime we introduce a new chapter, I think, whether it's anatomy and physiology, maybe zoology, and perhaps in this class as well, when you look at this, molecular genetics, what might that mean to you, genetics? Okay, put together what you two had said because you're both correct, just for different reasons. So what was it that you had said? What was it that you had said? Okay, so why are you answering for her?
Because you're not reading it.
Why? Well.
Because I was trying to get this down, and I was like wondering why it was so crinkled, and then I was like, oh, I didn't put it in a folder.
Well, that's going to happen. Okay, so from mom, then what did you say over there? Okay, so mom and dad gets built into you. That's what makes you who you are. Okay? Now, when it comes down to growing up today, obviously a lot of things have changed from the time that I was your age. I'm willing to bet that sometimes you're at home and mom and dad will say, well, back when I was your age, it was blah, 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 blah. Does this sound familiar? Okay.
Back when I was your age, if I talked to me, or if I talked to my mom the way you talked to me, I would have been dead.
Okay. So what do you think of that statement?
I mean, from the stories about my mom, I kind of, like,
Okay? So, because I have been asking other classes, okay? I don't know at what age. I don't think you can really answer this question. You may have thought about it, but if we turn the clocks ahead 10, 12, maybe 15 years into the future, some of you may be husbands, wives, mothers, fathers, any one of those. So what I want you to think about is, since we're talking about those genetics, do you want or would you like your son or daughter acting the way that only you can answer that. I can't say I wouldn't want them to be acting like you. You can answer that, but I can't. But do you want your son or daughter to act like you do now?
As long as they have big biceps.
Big biceps, huh? Okay. I think there's probably something a little more to it than big biceps. That's not where I thought it was going to come out when I heard the word big, but okay. Anybody else? So that's a self-reflection. Think of years ahead. And you could also look at it this way. Would you want your son/daughter to bring home someone to meet you as a parent?
They ain't bringing home no one. Period.
Okay. All right. So could be. All right. So this term genetics is going to mean something different for everybody involved. But typically what we mean, it's what you inherit from your mom and from your dad. Okay? In certain cases, I've always believed this. You play with the hand you're dealt. Okay? Has anyone ever heard that before? No one, the way the man upstairs designed us, we're not promised anything. Right? Would you agree with that? You have to, I think you get, sometimes you get out of life what you put into it. Is it fair all the time? No. But we don't get to make that decisions. Again, whether you believe it's just the grand plan of the way the man upstairs designed us, that's for you to answer. We're not trying to push that upon yourself. But everybody has something to add, I think, to make your life better, make other people's lives better. But I'm a teacher. I'm here to make your lives worse. How many people caught that?
I caught it.
Do you believe it?
Yes, 150.
I said, I'm a teacher. I'm here to make your life worse.
You're supposed to help us.
And we hope so. Hopefully, we feel that there is some benefit here. We are here to help you, whether it's myself or other staff members down the hall. Okay? So, moving forward then, okay? When we talk about, okay, how many of you like math? M-A-T-H.
Good at it, but I don't like it.
I've heard that before. A lot of students say, I'm good at it, but I don't like doing the math. Okay? There's a lot of things that we have today we've discovered because of math. Okay? And probably the most applicable type of math is not probably behind geometry. Well, I would say geometry and another specialized math are probably the driving force for everything that we see in the world today. And it's not algebra. Calculus, that's right. Calculus and geometry are probably the most applied mathematics. That's just my opinion. I could be way off with that, but again, I'm not trying to push an opinion upon yourself, but that's just what it is that I believe. Okay? I have some people sitting out there with some blank stares on their face. Okay? There was a note from last week. Who doesn't have, okay, we can't address that if we really don't know that. Okay. Yep. So if we call Gregor Mendel, the father of all genetics, it's actually Sir Isaac Newton is the father of mathematics, really. Okay, that of applied calculus. Okay. So eventually we're going to be reading about what this study is, and that's why you have your books, and that's why we're going to go through that here in just a couple of moments. So with that, we'll explain what this means here in just a moment as well. I don't think we're going to get as far as what I thought, but that's okay. So looking ahead a little bit, okay? Does anyone know what pneumonia is? Just tell me. Well, it can be a disease. It's from bacteria. Okay. Okay. It does, and it is. It's hard to breathe because a scientific term is called edema. It's called edema. That means you got fluid built up in your lungs. All right? So one way to look at it is as follows. Okay? What's the function of a cleaning sponge? You put it in what? Water, and it soaks up water, right? Okay, and the water belongs in there. Okay, there's lots of pockets inside that sponge that absorb that water. But your lungs, on the other hand, okay, typically when we cough, it could be from various reasons. One, you have something that's agitating what we'll call cilia in your trachea, that's your windpipe, but it's really called your trachea. Or if you inadvertently are about ready to take a drink, someone tells you something funny, you breathe in and swallow your water or whatever liquid it is at the same time, and then someone says, yep, it went down the wrong tube.
Or you just spit it everywhere.
And because you're expelling that. That's right. So it goes down into your trachea where it doesn't belong. If it really gets down low, it gets into your lungs, and then it starts to burn, actually. Yes?
This is from experience. Or it, you can swallow it, and then when you're laughing, it goes out your nose.
Yep, then you're really, that's a lot of pressure built up inside. And that burns too. Okay. But what happens is fluid is not meant to be in your lungs. There's supposed to be blood gases in there, oxygen and carbon dioxide. So kind of like that sponge, it's soaking up water, but in this case you want gas to be soaked up into your lungs, not water. Okay. We're going to talk about these strains of bacteria, and it's actually, I want to say, maybe phenomenal how someone can actually discover this, but something more that you can maybe relate this to is as follows. Now, you don't need to write this down, okay? But if you feel it's beneficial for you, well then write it down. But I'm not going to walk around and say you need to write this down. This is just something that's coming down the pike in the future. And what I mean by that is you probably have seen something like this before. This is where we're talking about what is inside you or what you get from your mom or your dad. And I'm sorry, what you said was right, but I can't remember what it was earlier. It's us. It's inside us. What was it?
It was
built into us. Okay. Okay. So when we're talking about eye color, okay, typically brown eyes are more dominant than blue. Okay? So what that means is, do you know for sure if your mom has brown eyes and your dad has brown eyes, does anyone born from that, will they have brown eyes?
There's like a low chance you get blue eyes.
Do that again. Why are you right? Because you're right. Jackson said no. No, that is not the case. That is not 100% true.
Make one of those tables.
It's called a Punnett square. You're right. Okay? So what that means then, chances are that if both of your parents have brown eyes, then the offspring will too. But the reason we say that is maybe maybe the dad has this eye color and the mom has brown eyes too, but they've got that blue-eyed trait. But is it possible, from what you know about this, just without any further course of action, do you think that this is brown eyes, or can there be some blue eyes in here? Maybe. Okay. So this is what we call a dominant trait, because we said brown is dominant over blue, right? Okay. But what can happen is both mom and dad do have brown eyes, but they've got that recessive trait in both of them. If one of them has the dominant trait and the other dominant trait, everyone's going to have brown eyes. Doesn't matter. Okay? But even if both mom and dad do have brown eyes, but they've got that trait in them, well, then there is that possibility 25% of the time that blue eyes can appear. How do we come up with the number 25%? Yes, it's in quarters, so 1 out of 4 chances gives you 25%. Okay? So this is how we figure out some of this genetics, through test crosses. You farmers, that is very important to you, genetics. The reason we know that is because I think you could almost ask any farmer now, the yields that they are getting from their corn and soybeans is probably better than what they expected. How's that possible? Because of genetics. That's why. Okay. So what we want to take a look at then is on or about page 326.
Are we done with this, Doohickey?
Yes. So around page 326.
Which page?
326. Okay. We should be there by now. Okay. Discovery of genetic material. It's written in blue there on page 326. That's approximately where we will start. Okay. So, once Mendel's work was rediscovered in the 1900s, because I believe Gregor Mendel lived in the early 19th century or 1820s, I believe. So he had done studies on pea plants, and we'll talk about pea plants as well, but typically when we get to that point, I'd rather talk about cattle, and that's no bull. Huh? 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 No bull? Huh? Huh? Okay, no bull. All right. So scientists began to search for the molecule involved in inheritance. Scientists knew that genetic information was carried on the chromosomes in eukaryotic cells.
Sorry.
She had to find a pencil. She's a little.
Okay.
I'm a little incompetent today.
So it's actually carried on chromosomes. That's why we say it's what you get from your mom and your dad. That pertains to this as well. Maybe eye color is on, there's 23 different chromosomes, okay? That's a haploid number. You put them together, you get diploids. So there's 46 chromosomes, 23 from mom, 23 from dad. Maybe this is chromosome number 8, and this gene is found on chromosome number 8. I don't know. I'm just using that as an example. That genetic material is found on chromosomes, and that's why you get it from your mom and your dad. Okay. That the two main components of chromosomes are DNA and protein. For many years, scientists tried to determine which of these macromolecules or nucleic acid, DNA, or proteins was the source of genetic information. So the scientist Griffith, okay, when you look on the right-hand side or on page 327, you see him doing studies with that of mice, okay, because he would inject them. Some of this bacteria was live. Some of this bacteria was heat killed, and then sometimes he mixed the strains and still killed the mouse. So why would something like that happen? So that's why we're going to describe that right now. So Griffith, okay, the first major experiment that led to the discovery of DNA is the genetic material was performed by Frederick Griffith in 1928. Griffith studied 2 strains of bacteria, okay, Streptococcus pneumoniae, which causes pneumonia. He found that one strain could be transformed or changed into the other form. And why would that be? Even though you heat kill a substance, how is it transforming into another strain? That's probably the question that he was asking himself, and that's what we're going to talk about here right now. Of the 2 strains he studied, one had a sugar coat. That's one of the very last things that said on your slide, right? It said one had a sugar coat on it. And that's what we're talking about right now. And one did not. Both strains are shown in figure 1. The coated strain causes pneumonia and is called the smooth strain. The non-coated strain does not cause pneumonia and is called the rough strain, because without the coat, the bacteria colonies have rough edges. That's why it's called the rough strain, so it makes sense. So follow Griffith's study described in figure 2. Notice the live S cells killed the mouse in the study. So that means that's the smooth bacteria, the S cells. The live R cells did not kill the mouse. And the killed S cells did not kill the mouse. However, okay, when Griffith and the killed S cells did not kill the mouse, okay, I'm sorry, that was the same sentence. However, when Griffith made a mixture of live R cells and killed S cells, remember, the S cells is probably what's going to kill the mouse. And that's what they're talking about here. Okay? And injected the mixture into a mouse. The mouse died. Griffith isolated the bacteria from the dead mouse. When these isolated bacteria were cultured, the smooth trait was visible, suggesting that a disease-causing factor was passed from the killed S bacteria to the live R bacteria. Griffith concluded that there had been a transformation from live R bacteria to live S bacteria. This experiment set the stage for the search to identify the transmitting substance. So, in short, You calling your shot there, Babe? Babe Ruth? That's why they can say the shot, because he's calling his home run shot. But anyway, page 327. So picture A, it shows you a picture of that dead mouse. I think, is there a caption for this? Yeah. Okay. So in letter A, it says in the smooth strain, the disease-causing substance was injected into the mouse. If it's disease-causing, it's no surprise, it's going to kill the mouse. Pretty straightforward. So letter B, the rough strain, the not diseasing-causing mouse, he injects that into the mouse, and it lives, okay? It's got that rough strain, or I believe it has the sugar coat. So that gets injected into the mouse, and it lives. No one can argue with that either. Well, what happens if he takes this disease-causing substance, or that S strain? There's two ways you can kill bacteria. One is with an antibiotic. The other is with heat treatment. So there's a reason why your internal temperature starts to go up when you get an infection. Trying to do is kill that bacteria off, okay? So that's why fever is just a normal reaction to try to combat or fight that bacterium. So what we see happening in letter C is that deadly strain was then, it says heat killed the S strain, okay? Then he injected that into the mouse lives, okay? Must be doing a study back there.
Yeah, I'm trying to look for what those things.
Oh, okay. And then in letter D, okay, so even though that strain was heat killed, does everyone see what I mean in letter D? Because typically that should not kill the mouse because that disease-causing bacteria was killed. But it's mixed with that of the live R bacteria, which is not lethal. So for some reason, that type of hybrid, when they mix together, the rough and the smooth, even though one's disease-causing, been killed, the non-disease-causing bacteria is still alive, those two, when they mix, for some reason, that kills the mouse. Well, it's the two type of bacteria that then reproduce to cause disease or probably pneumonia in that mouse. Okay? So that's what this is trying to show you, that even if in genetics, if you think you have the answer to this, sometimes there's something else going on when it comes to genetics. Whether it is brown eyes, we know this now. We know this with mice now, but think back to the 1920s or 1800s. Why am I mixing a tall plant and a tall plant and they're all tall? Why am I mixing a green pea plant with a green pea plant, but some of them are coming out yellow? Well, we didn't know that at that time. So studies like this would hopefully start to explain what's actually going on. This is a different type of math, probability or statistics. So when we say 1 out of 4, that's a probability, 25% of the time. Now, when it comes to this disease-causing bacteria, there's really no set number on percentages that they're trying to illustrate with that. But for some reason, the types of bacteria, even though it doesn't cause any disease, it mixes with a disease-causing bacteria and kills it, even if it's been dead. So something kind of strange going on. So I think that is enough for today. Tomorrow I'm not here. We have the Region 2B meet. I'll have a presentation for you on the Science Channel, and you can move forward with that. All right, we'll catch up to you next time.